So, make it like this. Hello everyone, my name is Suleiman Ahmed, and today my presentation is on the topic the glycogen metabolism in liver and muscles. So let's start. So something about glycogen. So what is glycogen? Glycogen is a storage form of glucose. It's actually a polysaccharide, and also it's what is called the energy storage, and the energy storage of the glycogen is in two forms. It can be a short-term storage or a long-term. So long-term, it can be a body fat. So that was all about the glycogen, because it's not our main topic. So this is the glycogen, and glycogen, it is formed by 1,6 bond or 1,4 bond. Depends, either it is straight or branched. Yes, that's all.
Okay, I show one.
Okay.
Because today in the quiz was question about the product of predominant product, yes? Only those glucose which are signed as green, yes, are released during degradation of glycogen as free glucose, yes? Because here we have this alpha 1,6 glycosidic bond, yes? And glycosidase remove free glucose. You can see that predominant product from all those gray glucose residues form glucose 1-phosphate. Yes, so this is a big difference between glucose and glucose 1-phosphate. Predominant form is glucose 1-phosphate.
So
This is a
shall
picture
we
to explain
No.
it.
So, okay, like this. So, about the glycogen metabolism. So, when we're doing the glycogen metabolism, we have to make sure that we are regulating it carefully, so that we can have sufficient glucose for the body energy needs. And there are like different hormones which contribute to glycogenolysis and glycogenesis, which are actually the main processes in the glycogen metabolism. Glucagon, insulin, epinephrine, norepinephrine, adrenals. These hormones, they promote to glycogenesis and glycogenolysis. Okay, they control it. Okay, is it fine? Next thing. So what things do we need for glycogen metabolism? There are different hormones, there are different proteins, some activators like CAMP. CAMP is cyclic AMP, which stimulates or activates the phosphorylase kinase, and then phosphorylase kinase in turn activates the main enzymes of the glycogen metabolism. I will discuss it later in the next slide, but I'm just giving a brief description about it. And about the protein kinases and protein phosphatases. These protein kinases phosphorylate. So it phosphorylates the glycogen enzymes, like taking part in the glycogen metabolism. And the protein phosphatases dephosphorylate. Their phosphorylation, their role is very important. Like, to one enzyme, the phosphorylation can make that enzyme active, or to other enzyme they can make it inactive. And about the insulin, glucagon, epinephrine, as I said, and we can also include the norepinephrine and adrenaline. Like, they just contribute to glycogenolysis and glycogenolysis. Is it fine till now? Okay. Now we'll just come to the main topic. Regulation and metabolism of glycogen in both muscles and liver. Oh, favorite picture. So, this is everything, but I will explain it. And there are some mistakes I made while I wrote, but when I will explain, I will just tell it correctly. So let's begin with the two main enzymes which are involved in the glycogen metabolism. So you see this glycogen phosphorylase and glycogen synthase. These two enzymes are the most important ones in this metabolism. And the process of glycogenolysis, which break down the glycogen into glucose 1-phosphate, is controlled by this glycogen phosphorylase. Okay, and the process of converting glucose 1-phosphate to glycogen is controlled by this enzyme glycogen synthase. So how is it happening? First I will begin with what's it called. Yeah, it's so much I have to see where to start from. Let's start from the example glucose 6-phosphate. Let's say a body has increased amount of glucose 6-phosphate. What does that mean? Increased amount of glucose 6-phosphate means that you have increased blood glucose level, right? Increased blood glucose level will, if you have increased blood glucose level, you don't need any more glucose. So what you want is your more glucose to be converted to glycogen. So for that, this glycogen synthase enzyme will be working. It should be stimulated, and on the other hand, this glycogen phosphorylase should be inhibited. It's just a general, it is how it should be working. Now I will tell how and who makes these things stimulated and inhibited. So let's say we have, as I said, we have increased amount of glucose 6-phosphate. This glucose 6-phosphate will have more, lead to more blood glucose level in body. This glycogen synthase, okay, sorry, I'm confused how to start. Glycogen synthase, okay, as I said, this increased amount of glucose 6-phosphate will stimulate glycogen synthase, but it will inhibit glycogen phosphorylase. If we have increased amount of glucose 6-phosphate, it means we have more glycolysis going on. Which means we are making more ATP via Krebs cycle. So if you have more ATP, our body doesn't need that much ATP, because the excess of ATP is also something that we don't need. So the ATP will also inhibit the glycogen phosphorylase as do the increased amount of glucose 6-phosphate. But there is one thing more. Free glucose level. If you have increased free glucose level, I'm not talking about the glucose 6-phosphate. If you have increased free glucose level, this can also inhibit the glycogen phosphorylase, but that's only in the liver, not in the muscle, because we only have free glucose level in the liver. And the third thing I have to mention it, but these three things, free glucose level, increased amount of 6-glucose phosphate, and the ATP, is inhibiting this glycogen phosphorylase. So what is stimulating it? Something that is stimulating glycogen phosphorylase is the AMP. But that's only in the muscle. AMP is the product when your muscle is contracting, skeletal muscle is contracting, and during the cross bridges you need more ATP to break down the cross bridge between the myosin and troponin. So actin and myosin. So that time you need more ATP. But ATP is converted to AMP. Increased amount of AMP stimulates the glycogen phosphorylase so that this glycogenolysis process can start over, and glycogen is broken down to glucose. That's what we need. Glucose, when we have glucose, we have glycolysis going on, Krebs cycle, we get ATP. So this is what happens. So let's begin with this process of this process. If we have decreased blood glucose level, first of all, I will just say before I go to there that we have two kinds of regulations: allostatic regulations and hormonal regulations. So what I mean by allosteric regulation is these enzymes, the glycogen phosphorylase and glycogen synthase, have allosteric site, and they are either stimulated or inhibited by the different molecules. And about the hormonal regulations means that the hormones come and bind and play their role either in phosphorylating or dephosphorylating. So let's talk about the allosteric. So something that I explained, like the increased glucose level, which stimulates glycogen synthase, and on the other hand it inhibits the glycogen phosphorylase. These were all the allosteric effectors, right? These are all allosteric effectors. And we also have one more thing over here to say, that protein kinase. Okay. So who is controlling this glycogen phosphorylase? Glycogen phosphorylase. Like what I mean to say is, glycogen phosphorylase is converting glycogen into glucose. How is this happening? There is another enzyme which is controlling this glycogen phosphorylase, which is phosphorylase kinase. And there are two forms of phosphorylase kinase: B form and A form. B form is inactive form of phosphorylase kinase, and A is active form of phosphorylase kinase. So what happens is, here in the glycogenolysis, phosphorylase kinase B is changed to phosphorylase kinase A. How does it happen? For this, I have to go back and start over with this so that I can relate how this will happen. This we have a G protein coupled receptor, and here in G protein coupled receptor, if you have low blood glucose level, so which hormones will get stimulated? Your glucagon will stimulate it, your epinephrine, your norepinephrine, your adrenaline can also be stimulated. So it can be any. So when you have low blood glucose level or when you are stressed, in both cases, your either one of the like hormones that I just mentioned, just say epinephrine. Epinephrine will bind to this G protein coupled receptors. After it binds, it will stimulate the G stimulatory protein. G stimulatory protein will bind to GTP, and this G stimulatory protein will become active. When G stimulatory protein become active, it will go to the enzyme factor, which is the adenylate cyclase. Adenylate cyclase will pick the ATP and convert that ATP into cyclic AMP. Cyclic AMP will stimulate the phosphorylase kinase A, just that I mentioned in the last slide. Oh, you're right. So CMP will stimulate the phosphorylase kinase A, and here it goes phosphorylase kinase A. So sorry. This is not phosphorylase kinase. This is protein kinase. This is protein. I just confused. Yeah. Yeah. So this CAMP has stimulated the protein kinase A. Protein kinase A came here to the phosphorylase kinase B and phosphorylates it. So what does, as I said in the last slide, protein kinase A function is to phosphorylate. So it will phosphorylate the phosphorylase kinase B, and so when it will get phosphorylated, it will change into its active form, which is the phosphorylase kinase A, and this phosphorylase kinase A will in turn phosphorylate this enzyme glycogen phosphorylase, and then it will be stimulated, and this process of glycogenolysis, which means converting degradation of glycogen into glucose 1-phosphate, will take place. And that's what we wanted, because we had low blood glucose level. So what we needed was increased blood glucose level. So this was a process, and we made the increased glucose level. But there is one more process. There is one more process which can do this all, which is the other process is like done by the different G protein coupled receptors. For that, epinephrine and norepinephrine, these two bind to the G protein coupled receptors, and stimulate the GQ, stimulate a different protein, which is GQ protein. When GQ protein, it's the same process as here. GS protein binds to GTP. Here also GQ will bind to GTP and become active, and this GQ protein will go to PLC, phosphorylase lipase C, and make it active. This phosphorylase C will do one thing. What will it do? It will break down the PIP2 into DAG, which is diacylglycerol, and into IP2. I just made it wrong, maybe because I was sleeping while I was doing it. It is IP2 like IP3. So PLC broke down the PIP2 into diacylglycerol and PIP3, which is triphosphate. Okay, this PIP3 goes on the receptor on the endoplasmic reticulum, and you know what we have in the endoplasmic reticulum? Calcium ions, right? So this IP3 binds on the receptor on the endoplasmic reticulum and pulls out the calcium ions. Those calcium ions play an important role. Those calcium ions bind to a protein known as the commogulin, and the complex of this calcium commogulin protein will phosphorylate what? Will phosphorylate the phosphorylase kinase B. When phosphorylase kinase B is phosphorylated, it will change itself to active form phosphorylase kinase A, and again the whole step will repeat. Phosphorylase kinase A will in turn phosphorylate the glycogen phosphorylase and express itself so that glycogenolysis can happen one more time, and glycogen is degraded to glucose 1-phosphate. Oh, holy shit, that was a lot. So, but there is one other thing as well. If this protein kinase A is phosphorylating the glycogen phosphorylase, it is not just what's it called? What's the word? I'm forgetting. This protein kinase A doesn't just phosphorylate this glycogen phosphorylase. It is not just specific for this glycogen phosphorylase. It will also go and phosphorylate the glycogen synthase. Before I just explain it, the process, I'll just tell you that glycogen synthase has also two forms: glycogen synthase A and glycogen synthase B. A means it is active and B is inactive. So when this protein kinase A will phosphorylate the glycogen synthase, it will turn into the inactive form because it is phosphorylated. So we can say that protein kinase A, when it phosphorylates the glycogen phosphorylase, it becomes active and expresses itself, and glycogenolysis happens, and glycogen is broken down to glucose. But when this protein kinase A phosphorylates the glycogen synthase, it is inhibited, and the process of glycogenesis, which is converting glucose 6-phosphate to glycogen, doesn't take place. So is that good, right? So now I will explain the other hormone, which is insulin. When do we have insulin? Guys, we have insulin when there is increased glucose level in blood. At first I explained when we have low blood glucose level.
Increase.
Yeah, increase, you're right. And I said the same thing. So I'm saying before I explain when we have low blood glucose level, and we did all the process to make the glucose. Now we have high glucose level. Now the body doesn't need that high glucose level. It will make sure that extra glucose is converted to glycogen. So this glycogenesis process will be stimulated by glycogen synthase. How it will stimulate it? I will tell you. Increased glucose level will stimulate which hormone? Insulin, right? Insulin will bind to the special receptor, which is tyrosine kinase receptor. Tyrosine kinase receptor will stimulate the PPP, which is phosphoprotein phosphatase. This phosphoprotein phosphatase, when it will go to the glycogen synthase, it will pull off all the phosphates that were that were added by the protein kinase A. When it will pull off when this PPP phosphoprotein phosphatase will pull off the phosphates from the glycogen synthase, what will happen? This glycogen synthase will become active. We will have active form of glycogen synthase, and this will lead to the this will stimulate this glycogen synthase, and obviously glucose 6-phosphate will be converted to glycogen, and glycogenolysis will be taking place. And on the other hand, the protein phosphatase will also take off or pull off the phosphates from the phosphorylase kinase. When it will pull off the phosphate from the phosphorylase kinase, it will make this inactive, and then inactive you don't have active form. Then protein phosphatase will go to the glycogen phosphorylase, pull off all the phosphates and make it inhibited. Then there won't be a process going of glycogenolysis, only this glycogenolysis will take place. So that makes more sense. Initially we had high glucose level, so what we wanted was to convert extra glucose into glycogen. So you see, glycogenesis is like happening, and the other process of glycogenolysis is not, like, it's inhibited. And apart from this, like apart from the glycogen phosphorylase, which is taking part in the glycogenolysis, there is one more enzyme which contributes 1 to 2% of in glycogenolysis. That enzyme is known as lysosome, but the special name given to it is acid maltase or alpha-1,4-glucosidase. What does it do? It also does the process of glycogenolysis, but if we have deficiency of this enzyme, it can lead to significant problems, especially the heart problems. And there is one what's it called, glycogen storage disease, which is related to deficiency of this acid maltase, that is Pompe disease. Pompe disease is the glycogen storage disease type 2. So if I have discussed this, so I will just tell some more like important the diseases, glycogen storage diseases. There are actually 6. So how I remembered it, it was like, I will just tell you. First, there is von Gates disease. This von Gates, each of these disease is due to the lack of some enzyme. Von Gates disease is due to lack of glucose 6-phosphatase. Second, Pompe's disease. Pompe's disease, as I just said, Pompe's disease is due to the lack of acid maltase enzyme. Third comes the Coris disease. So Coris stands for starts with C. What comes after C? D, right? So just to remember, Coris enzyme is Coris disease is due to lack of the D branching enzyme. So Coris C, what comes after C? D. So Coris disease is because of deficiency of D branching enzyme. Then comes the Anderson disease. What comes after A? B, right? So Anderson disease is because of the lack of the branching enzyme. Then the last two, McArdle's disease and Hirsch's disease. McArdle's, as it starts with M, this is due to these two are due to deficiency of the glycogen phosphorylase, but McArdle's, as it starts with M, it is deficiency in the muscles of the glycogen phosphorylase. And Hirsch's disease, you know, the Latin name of liver is hepatic, starts with H. So just to remember easily, like it makes it easy, H with liver. So Hirsch's disease is also due to the deficiency of glycogen phosphorylase, but only in the liver. So this was how it was like numbered. So von Gates disease is glycogen storage disease 1, type 1. This Pompe's disease type 2. Coris disease type 3. Anderson disease type 4. McArdle's disease is type 5, and the last Hirsch's disease is type 6, glycogen storage diseases. And that was all that I wanted to explain from here. So let's move on. But before going to the next slide, I'll tell one more thing. That's a conclusion thing. So glucagon, epinephrine, and norepinephrine, or adrenaline. They stimulate the process of glycogenolysis, breaking down glycogen into glucose 1-phosphate. The other hormone, insulin, stimulates the process of glycogenesis, making more glycogen from extra glucose 6-phosphate. Glucose, 1-phosphate. So let's move on. Now, differences between muscles and liver glycogen. If I talk about the differences, the very basic, the main difference between the liver and muscle glycogen is in the muscles does the process of glycogenolysis so that they can produce more ATP via glycolysis and Krebs cycle. But liver does the process of glycogenolysis, same process, just to maintain the blood glucose level. When you have low blood glucose level, this liver does this process of glycogenolysis so that it can make more glucose, and that glucose is released into the blood. And in the liver, we have an enzyme known as glucose 6-phosphatase, but in muscles we don't have such enzyme. And one more thing, in muscles, AMP is something which is the allostatic activator of these glycogen phosphorylase. On the other hand, in the liver, glucose is the activator of the glycogen phosphorylase. That means if we have AMP, the glycogen phosphorylase will start the process of glycogenolysis, breaking down glycogen into glucose, and by that way you will have more glucose, right? And on the other hand, in the liver, this AMP won't work. You need glucose over there so that you can activate the process of glycogen phosphorylase. Okay, that's all. And this is something same as I explained. This is a diagram which is showing you that in liver glycogen is broken down to glucose, and glucose is released into the blood. And here, because we have an enzyme in liver, we have an enzyme glucose 6-phosphate, phosphatase, which will change glucose 6-phosphate to glucose, and that glucose is released in blood. In liver, in muscle, what do we have? We don't have this glucose 6-phosphate, right? We don't have. But what we can do? If we don't have it, so glucose 6-phosphate will stay glucose 6-phosphate, and it will undergo the process of glycolysis and Krebs cycle. If we have aerobic respiration, then it will make ATP. That was all. Thank you so much. I hope you enjoyed the video. I mean, not video, presentation. Okay, thank you.